All right, so welcome to the final exam review. This is our last lesson as a class. You do have your labs this week to continue review, but this is your last sort of, I guess, lecture, if you want to call it. Um, so we'll go through the whole second half of the term because that's what's on the final exam. And after your break, we started with nutrient deficiencies. Remember, one of the key things for nutrient deficiencies was to determine if the symptom was from a mobile or immobile deficiency or mobile or immobile nutrient. In other words, if it's immobile, that means that we cannot relocate the fertilizer or the nutrient to the top of the plant. And if it's mobile, it means that the plant can extract the nutrient from old tissues and recycle it. So we would expect to see immobile deficiencies at the top of the plant and immobile ones at the bottom of the plant. So breaking it down into partially mobile or mobile principally, the best way to look at this is that your macronutrients, for the most part, are mobile. So NPK, magnesium, and sulfur kind of fits into that macro. It's a secondary macro. And then the only micronutrient that's mobile is molybdenum. Now, typically, to help you remember, for non-mobile nutrients, your micronutrients are principally your non-mobile, except for calcium. Calcium is the only secondary macronutrient that is immobile or non-mobile. So these deficiencies would all these present at the top of the plant. Okay, so some examples and terminology. Chlorosis means that we've lost chlorophyll, so the plant becomes yellow. Sometimes we call the leaf chlorotic, meaning that it has lost its chlorophyll, has become yellow. But the process of losing chlorophyll is called chlorosis. Interveinal chlorosis means that we've lost the chlorophyll between the ribs and veins in the leaf. Okay, that's an important term to use, interveinal chlorosis. Necrosis is death. So see these brown edges of the leaves? That is necrosis, dying tissues. Typically associated with later stages of a deficiency like potassium. That is the term we would use. So you might say that a leaf is necrotic and that it is dying, or it is displaying necrosis. So here we have a young strawberry leaf, and it has what on the edges? What do we call that? Dying tissue. It has necrosis at the tips of the leaves. So it is a young leaf at the top of the plant. So it would be a immobile nutrient, and we'll call that a calcium deficiency. Calcium is responsible for growing healthy cell walls, and if the plant can't not get calcium to the tips of the leaves, which is the furthest from the roots, are the last ones to get the calcium. So the cell walls are weak and the cells die and dry back. So that's common calcium deficiency. Here we have corn, which is a monocot. We're looking at older leaves, which have yellowing and necrosis on the edges. This is associated with potassium deficiency. Okay, so potassium starts with yellow margins, and as it progresses, it quickly turns into necrosis. Here we have old leaves on tomato plants. So they're old, not new, which means that it is a mobile nutrient deficiency. And intraveinal chlorosis is often associated with a magnesium deficiency. So we would give these tomatoes Epsom salts to fix this deficiency because it gives the plants magnesium sulfate. Here we have a plant that has yellow on the apical leaves. This means that it is an immobile nutrient because it cannot move the nutrient from old leaves to the top. So it's immobile. And any kind of yellowing at the top of the plant that is completely across the leaf is typically an iron deficiency. And of course, as I've talked about in class, with an iron deficiency, the first thing you want to check is pH before you add iron. Here we have a young cannabis plant, and it is yellow at the apical part of the plant, the newest growth. So this nutrient is it mobile or immobile? It is immobile because the youngest leaves are displaying the symptom. And in this case, it would be an iron deficiency. Again, iron is related to pH, right? So as long as we have an acidic pH, the iron is available. Here's a picture of blossom end rot in tomatoes. This happened at a very young stage during the flower, and it is related to what nutrient? Now, typically, it is immobile nutrient, and it is a secondary macronutrient, and it is calcium. Okay, calcium is responsible for the structural integrity of cell walls, and the very end here of the tomato is the very end of the flower. It's the furthest part from the vasculature, so it's the last part to get calcium, and so it dies. And as the tomato grows, that end, necrotic end just gets worse and worse and worse. Here we have this is a canola or mustard plant, and it is exhibiting deficiencies on the older leaves, which means it is an mobile nutrient, which means that whatever is deficient in the plant can be extracted from the old leaf, moved up the vasculature to the top of the plant where it's prioritized. So typically, older leaves turning yellow is indicative of nitrogen deficiency. Here we have purpling under the leaves and purpling on the petioles. Purpling is associated with what nutrient? Notice it's not purple at the top of the plant. It's purple at the bottom of the plant. So it is mobile. So what the plant is doing is it's extracting phosphorus from the older leaves and prioritizing it to the new leaves. So it's a phosphorus deficiency. In class, we talked about Liebig's law and how the growth of a plant is limited by the singular most limiting nutrient. In this case, nitrogen limits the entire growth of the plant. So oversupplying one nutrient isn't going to compensate for whatever is deficient. So the plant its full potential is limited by the most single limiting element for growth. We also talked about the rhizosphere, which is in the soil and the influence of the roots. So it's around 2 millimeters around the roots. That immediate area, it has high biological activity. Remember, the plants have a root cap. It's sloughing off cells all the time. And the roots are also producing mucilage to help them move through the soil. All these things feed bacteria. So the immediate vicinity of the roots, let me just clean this up, always has lots of biological activity. Okay, all the bacteria right around the roots are feeding on these exudates and sloughed cells, and that means that this rhizosphere has lots of biological activity. In terms of decomposers, we talked about first fungi being principal for late stage decomposition, breaking down mostly cellulose. We talked about how what we often see is just the fruiting body, and it's a much smaller representation of what's actually there. This entire log is full of hyphae, and the few little things that we see, mushrooms or whatever, are just a small reproducing structure. The hyphae or fibers would penetrate throughout the tissue as they break down the cellulose and lignin. And we also introduced you to mycorrhizae, which are a fungus that, in addition to breaking down cellulose, also enhances nutrient uptake like nitrogen, phosphorus, and copper. It also helps bring water to the plants in exchange for sugar. So what we have here is a root structure. So this would be the plant root, if you can see that, right? That's the plant root. And all these other structures that are happening around here are mycorrhizae growing out from the roots, enhancing the plant's ability to collect more nutrients and water. In soil biology, we talked about bacteria. These are the heavy lifters. Basically, anything that's fresh, they decompose. And there's, round and rod shaped ones. We don't get into that too much. But the bacteria are principally for fresh. Remember that for your exam. Fungi are for old, mostly cellulose and lignin. The bacteria eat the freshly uh, dead or whatever decomposing plant material. Um, Green, if you want to talk composting. I also introduced you to actinomycetes, which are a beneficial fungi that, fungi, it's kind of a crossover species between bacteria and fungi that exists in the soil, and it produces antibiotics. And they will protect plants from diseases and also enhance the breakdown of cellulose and give the soil a wonderful earthy smell. You're smelling molecules that the actinomycetes is producing. That's in the late stages of decomposition, actinomycetes. And in your labs, we got to see protozoa that eat the bacteria, consuming them, and then releasing the nutrients that they have mineralized. There's that word we talked about in the last week of lessons, and here it is again. Mineralization is the process of breaking down complex plant and decaying matter into the individual nutrient elements like nitrogen, phosphorus, and potassium. And there's lots of different protozoa, flagellates, amoeba, ciliates. Um, we also introduced you to the idea that where there's grassy areas, so fields and stuff like that, are predominantly bacterial because mainly it's a lot of fresh green material, not a lot of dead. And in the forest, there is mostly fungal. So this is partly why we mulch trees in a turf setting, because we want to have the beneficial fungi for the tree and we need the bacteria for the turf. So little interesting kind of tidbit on the differences in soil for different contexts. So again, in the forest, fungi are breaking down leaf and wood material, um, and bacteria are for the fresh material. Now, as far as whether it's composting, vermicomposting, or soil biology, the principles are all the same. The factors that affect quality are things like aeration, which is oxygen, right? Oxygen. And moisture or water content, temperature, food source, pH, and also limited competition is helpful, right? So we don't have too many diverse things going on. And of course, the soil texture helps as well. So if it's really coarse, that will be not as good as a finer. We talked about compost tea, the idea that you start with some compost or healthy soil and suspend it in water. And the key here is vigorously aerating. We do not want anaerobic conditions. And then we wait 24 hours. And then all the bacteria and everything that has reproduced and formed a dense population, we take that and we add it to our soil. And that enhances the bacterial population and accelerates the mineralization of decaying material into plant food nutrients or whatever. So with oxygen, obviously we want to avoid anaerobic conditions because that leads to bacteria that actually they consume the nutrients and then there's nothing left for the plants. Now I know that in some situations you run into things like bokashi, which is a fermenting style composting process. That is complex and we're not going to really get into it too much. But generally speaking, we want to avoid anaerobic conditions. We don't want anaerobic conditions. We want lots of oxygen. And one of the ways that we can assess if the compost tea has been made properly is to look at the types of populations that we see. Now, this doesn't mean that it is anaerobic on this side. It just means it is less aerobic. So in your labs, we saw quite a few paramecium. And of those of you who got to see slides from compost tea that was left in the lab for 24 hours again, you notice there is a lot more paramecium because they are much more diverse and they can survive with less oxygen. The more oxygenated compost tea would have more amoeba. Now, in the lab, we saw mostly testate amoeba. That's these things in a shell. So you sort of see a round shell, and then there's a soft part kind of coming out. Didn't see a ton of them, which tells us we're not getting as much aeration as we thought we were. Then we introduced you to the concept of vermicomposting. So we've got composting, and then we've got vermicomposting. With vermicomposting, we are using worms to decompose all the various ingredients into nutrients. And that's just basically worms digesting. And inside their gut are bacteria that further mineralize the nutrients. Now, by comparison, we have conventional compost and we've got vermicompost. And with conventional compost, there is roughly only 10% of fresh material can be added. And in vermicompost, up to 50% material that is fresh or green can be added. Remember, in compost, it's 30 to 1 carbon to nitrogen, C to N. It's really hard to write with my mouse. I've never gotten good at it. As far as compost goes, it requires extensive aeration to achieve rapid decomposition. So commercial facilities actually pump air into the compost, where vermicompost, you don't need that energy level. The worms take care of it themselves. With compost, we get high temperatures, enough to reduce pest and disease, where with vermicompost, it's room temperature, so we do not pasteurize the soil. So vermicompost will not kill weed seeds or pathogens inside. So we got to make sure we don't put any diseased material into vermicompost. So conventional compost is basically an amendment to soil. It's considered a slow release, and it increases soil properties like water holding capacity. Where in vermicompost, it's a much more intense nutrient load because those bacteria in the gut have mineralized more nutrients. It has far more nitrate, which is beneficial for nice, firm, healthy growth, not soft growth. Also, some of the bacteria can protect against diseases. For example, we have this link here that was to a paper that talked about compost tea being used to prevent pythium in seedlings. As far as the benefits for vermicompost, it's structural. Here you can see sort of the nodules that form, which create sort of core structures. So there's micropores and macropores. Definitely a soil enhancer. And again, negatives, there is no pasteurization with vermicompost. We don't achieve high temperatures. And we can only take so much in, right? Worms need time to eat it. Now, it's more than compost, but you still have to be careful you don't overload those worms or you start getting ammonia production. Vermicompost is, in fact, bacterially dominated, so we have to keep that in mind. As far as vermicompost goes, we have to use bedding that is going to absorb water. It will have good bulk, which means it will hold water but has air pockets so that oxygen can get in. And it has to have a high carbon to nitrogen ratio. The worms are getting their carbon from the bed and their nitrogen from the fresh foods. So we want lots of carbon in our bedding material. Horse manure, peat moss are good bedding materials. Coir and shredded newspaper are also very good. Even cardboard. There's some pictures of the different bedding type things. Red wigglers can eat about their own weight per day, although generally we try to stick to about half the weight. So if you have half a pound of worms, then you can put a quarter pound of fresh produce inside the layer at any one time. So looking at this cup of watermelon fittings, that might be about the maximum for one day that you can put into your layer of vermicompost. Otherwise, you're going to overload it, and you're going to start producing ammonia, which means that the nitrogen that you're producing will evaporate, and there'll be less nutrient quality in your vermicompost. We don't want that. Basically, if it smells bad, you're overfeeding it. It should have no smell whatsoever. Moisture content differs also between vermicompost and compost. With compost, typically we want a 30 to 40% moisture range, whereas with vermicompost, we want much more moisture content. It's going to be very wet. I don't know if you remember from the compost, we decided if you squeeze it hard, you get maybe one drop. That's how moist compost should be. With vermicompost, you can give it a good squeeze and there should be a dribble of water. So quite a bit more moisture. And that's primarily because worms breathe through their skin and their skin cannot dry out or they will die. Temperature is important. Basically, the lesson here is that worms like to be the same as us. So whatever we find comfortable, they'll find comfortable. If it's too hot for us, it's too hot for them. Too cold for us, too cold for them. Once they've done consuming everything and all the bedding is gone, so all the newspaper or whatever you've used for your bedding material, once that's been consumed, you stop feeding, wait about a week or two, and then you can put a new tray on top, make sure it's in contact, and you can start feeding the next layer. The worms will migrate up to the fresh layer. That's how you use your chalet, right? It's going to keep the worms will move up. When there's no more food, they'll go look for new food. If you're doing anything wrong, the worms will know because you'll see them trying to crawl out the sides. So worms will be exiting the trays, crawling out, trying to get away, thinking, oh my goodness, I have a terrible owner. um, Lastly, we discussed organic fertilizers. I tried to highlight the differences between conventional and organic production and conventional fertilizer and organic fertilizer. The key here is organic means it has carbon in it. So this molecule, whoops, missed one. This molecule has carbon in it. That makes it organic. It does have nitrogen. It's got nitrogen here. And that's about the only nutrient quality materials in this particular glutamine amino acid that would be in plants as they're decaying. So the plant would get two nitrogens out of whatever the results of this being materialized or mineralized by bacteria. Remember, we have things like bacteria feeding protozoa and animals and fungi all contributing to mineralization of inorganic components that plants can feed on, nitrogen, phosphorus, potassium, and things like that. We discussed the advantages of organic fertilizers, right? So that when you feed organic fertilizers, yes, in that previous slide, the glutamine, the only plant is only getting two nitrogens, but the other materials end up enhancing the organic matter content, which boosts cation exchange capacity in soil. They also enhance water holding capacity, improve structure, enhance bacterial populations, which will also accelerate more mineralization if you add more organic fertilizer. And there's some evidence that there's somewhat reduced leach um, of organic nutrients, mainly because there's just less fertilizer present with organic. So if it does, you have a lot of rain leaching it, there's less to wash away. The other thing is mycorrhizae, which we talked about already, have even better activity in organic context because there's nothing to damage them. So you get a lot healthier soil in that context. Disadvantages, and there are some with organic fertilizers, are that they are typically very slow. Oftentimes, what you're feeding now is for next year. Mineralization takes time, and the bacteria need the right temperature, pH, moisture, all the conditions for them to grow at a good rate in order to get sufficient mineralization to provide the nutrients that plants can actually eat. Temperature is a big slowdown with organic fertilizers. If you have cool conditions, your plants will struggle to get food from the nutrients because mineralization will slow almost to a halt in cool soil conditions. The other disadvantage is that you may not really know quite what the ratios of nutrients you are supplying at any one time. It's very difficult to coordinate that you get all nitrogen, phosphorus, potassium, whatever, supplied at equal rates all the time because different organic fertilizers supply different elements out of that, and they don't all supply it at exactly the same time. So that is a significant hurdle with organic production. So in summary, feeding with organic fertilizers enhances cation exchange capacity, increases water holding capacity. It relies on mineralization to produce the nitrogen, phosphorus, potassium, etc., etc., etc. All those nutrients have to be mineralized. They have to be produced through the process of mineralization. And that takes time, temperature, moisture, pH, oxygen, other factors that make the microorganisms happy. Anything that makes the bacteria happy is going to accelerate mineralization. So that's really critical. Typically, organic fertilizers are slow. You're talking minimum for the fastest organic 1 month, right? All the way up to 1 year in order for mineralization to happen. Growers don't always understand how plants will react to different organic fertilizers. Are you going to get stretch or soft growth? How's it going to impact the quality of the produce? Hard to say. We don't know all the science behind that. We do know that oftentimes it is healthier. We talked about that in the labs, but we know very little about this. Lots of claims are made, but the truth is we don't know. Definitely, when we talked about the different organic fertilizers, we want to be keeping food safety in mind and look at where did the organic fertilizer come from. Oftentimes, it's a waste product. Is it from a safe source? Are there risks associated with that source? Is blood meal from cows that are diseased? There are diseases that they can be passed on through that. So we have to be careful about the choices that we make in order to make sure that our organic fertilizers are indeed safe. All right, that's a quick summary of the term. We'll cover some more stuff in the labs, and we have some practice questions to go through to discuss. And in the lab, there's a Jeopardy game that we'll play too, which is kind of fun to practice and test your knowledge to see that you're prepared for the final exam. All right. Hope you enjoyed this quick lesson. Take care.